Welcome to Research Renaissance, presented by the Karen Toffler Charitable Trust. Embark on a journey into the forefront of cutting-edge brain science as we feature insightful discussions with a diverse array of guests, from early career researchers pioneering groundbreaking science to influential voices in investment communities, policymaking, and research institutions. Together, we delve into neurological diseases, their causes, treatments, and the quest for cures. Join us in advancing our understanding of the brain and addressing its ailments. Welcome to Research Renaissance. I'm Deborah Westphal, your host and guide through the fascinating future of the brain. Joining me today is Ashley Carey, a PhD candidate and a graduate research assistant in Dr. Silvia Facetti's lab at Temple University. Ashley is also a 2023 Toffler Scholar. Today, we will be discussing neurovascular dysfunction and neuroinflammation associated with Alzheimer's. So let's dive in. Welcome, Ashley.
Thank you for having me.
Yeah, I'm really excited. We haven't talked in maybe a year, and so I know there's been some fun things that you've been doing and kind of advancing your research, advancing your career. So I'm really looking forward to getting caught up with you today.
Yes, me too.
So let's just start with, what initially drew you to studying Alzheimer's, and, can you just share what any pivotal moments you had, and maybe just tell us why research?
Yeah, so I've always been more science forward in school. And I actually, for the longest time, I really wanted to be a marine biologist. But then all of a sudden in high school, I got introduced to the topic of neuroscience, which, during that time, it was an emerging research field. So I started to do more research into it. And then as soon as I went to Lafayette, I knew I was going to declare my major as neuroscience. And the whole time I really was just pre-med, but then I met a really awesome professor at Lafayette College, Dr. Reynolds, and she taught me all about the research path. She actually let me be an intern in her lab, so she showed me the workings of how a laboratory operates. And I just changed completely at the last minute and was like, okay, research it is. And I applied to a program, and then I ended up at Temple, where I really learned what Alzheimer's disease was. I learned about neurodegenerative diseases in general in college, but really the actual foundation of dementia, I learned all of that information working in my PhD lab. Yeah.
That's interesting. So marine biology and, the study of animals, and then now you're studying people and people's brains. So kind of what is that relationship there? And because it seems like that would be maybe an easy jump. I don't know.
It's definitely a little bit different, but just kind of learning how the brain really does control every other organ system in the body. Neurons project from your brain all the way down to your feet. So, really how neurons communicate with each other, how they communicate with other organs. That's really what drew me into neuroscience. And also, if your brain fails, your whole body is not going to operate properly. So it's kind of a really cool, the fact that the brain controls so much of, our daily activities.
Yeah. So when you got into your graduate studies, what was the most kind of surprising thing about the brain that you learned?
Well, definitely in college, I really only learned what neurons were. I didn't really learn that there's other types of cells in the brain. You have your glial cells, which kind of do some immunosurveillance. And then also the vasculature. I feel like when people think of the brain, they think neurons, but there's a whole network of vasculature, and that's really the lifeline to every cell in the brain. So it was just really interesting. There's a whole other side of neuroscience, which is the vascular portion of it.
Yeah, and you're in Dr. Facetti's lab, which is really, and I had her on the podcast not too long ago, and that is really kind of that vascular, blood, and then brain. What is it like to be kind of in that lab? Because she talked about some of the cross functions, some of the different kind of areas of studies.
Mm-hmm.
What is that like to be in her lab, and what is your specialty?
So being in the lab is very interesting. There's a lot of projects going on. There's a lot of people, but it just shows very how broad the field of dementia can get, even when you're focusing specifically on the vasculature. So everybody is focusing on either, you have somebody looking at heart function in Alzheimer's disease or specifically neuroinflammation, other people looking at blood-brain barrier permeability. So you really get to learn through all these projects, all the different subsets of kind of dysfunction that could be associated with, Alzheimer's disease.
And your kind of specialty is, or, the projects that you work on, what are those focused on?
So my thesis mainly focuses on looking at how cardiovascular risk factors impact Alzheimer's disease pathology and risk, specifically looking at diseases and risk factors such as hyperhomocystinemia. It's a condition involving excessive plasma homocysteine levels, which you can get from having deficiencies within vitamins within the body, and it causes a buildup of this metabolite in your blood, and it's very detrimental to not only your peripheral organs but also your brain. And then also I look at hypertension as well as a risk factor for Alzheimer's disease, and specifically how it impacts cerebral endothelial cells within the brain's vasculature.
What was that last word you just said? Cerebral what?
Cerebral endothelial cells.
Yeah, what are those?
So within vessels, there's different types of vessels. So for example, a bigger vessel, within you would have a basement membrane, and then around that you would have endothelial cells, and then around that smooth muscle cells. So the endothelial cells are really important for maintaining the integrity of the vessel, so nothing leaks out. And then also smooth muscle cells are important for the contraction and relaxation of the vessels. And then smaller vessels would have endothelial cells, but instead of smooth muscle cells, we would have pericytes. So both vessels have endothelial cells. So they're kind of a major component of all types of vessels in the body.
So, this is kind of a new area of study, this idea that there's this relationship between the vascular system and the brain. How fast do you think it's moving? It's fairly new.
Yeah.
But how fast do you think it's moving? Are we advancing that knowledge pretty quickly?
I think we are. I think a lot of people are starting more basic science. So trying to look at very specific molecular mechanisms that risk factors could, or due to the vasculature to progress Alzheimer's disease. But I think a big thing would be trying to find biomarkers to kind of identify these, something that we could use to screen. So if you have a risk factor, you could screen for this biomarker and see if you're having an increased risk for dementia. I think that's really where the field is trying to head with this. So that's a little bit slower, but I think a lot of good basic science is getting done, especially even in Dr. Fasadi's lab, we're really understanding how the cardiovascular system and its health can impact the brain.
How much do you guys study then the external? Because if you have risk factors because of genetics or, some of the, maybe just the things in life,
mm-hmm.
but there's other patterns of life or there's environmental aspects that also, how much do you kind of look at the environmental aspects?
Yeah, I guess these risk factors in part, they are genetic, but also lifestyle choices, I suppose. If these could be, high homocysteine in your blood could be caused also by, a poor diet, hypertension, also eating, too much salty foods or, not exercising enough, having too much stress. I think that all interconnects, back to kind of lifestyle choices. But we haven't really focused on other specific diabetes or anything like that.
Yeah. So the time that you've been in your graduate studies and with working with Dr. Fasadi, what are some of the kind of the significant findings that you've had from, some of the projects you're working and some of the risks that or some of the research that you've been doing?
So specifically focusing on hyperhomocystinemia, we just published a paper in Aging Cell, actually, And we found some really interesting findings within that study, particularly when we expose those cerebral endothelial cells that compose the vessels in the brain. When we expose them to amyloid beta, which is the main misfolded protein within Alzheimer's disease, and also high levels of homocysteine, we could see that there is an additive or a worsened level of endothelial cell dysfunction. So specifically, we see activation of cell death pathways, particularly something called apoptosis, which is programmed cell death, and also necrosis. Additionally, we found that exposure of the endothelial cells to homocysteine and amyloid beta also additively decreases the barrier integrity that these cells are able to achieve. So their barrier is more permeable, so it would allow for the leakage of, immune cells causing inflammation or other, bad bloodborne proteins into the brain. And also we found that this additive treatment also exacerbates proliferative or growing capabilities of endothelial cells. So, they're getting injured, but they also are unable to grow and form new vessels. So it's kind of just like a very vicious cycle with this amyloid plus this risk factor where you're getting continuous vessel damage and then the vessels aren't able to recover.
So, well, first of all, that's a really cool thing that you published in that. And I saw somewhere that it had, when it came out, it was one of the most downloaded reports of the year in that publication. Is that true?
Yeah.
That's a big deal.
Yeah.
Yeah.
I was excited.
Awesome. Congratulations for that.
Thank you.
Awesome. Yeah, that's great. So, and I don't know because I don't quite, of course I don't quite understand, but if we have drugs that can clear up some of the amyloid, is this your findings? Is there still such damage to the brain that, it wouldn't matter if the amyloid's cleaned out? Am I understanding that correctly?
Yeah, that's kind of so there's a lot of things that are happening actually before the amyloid is accumulating and causing significant damage. And that's where the whole vascular changes come in. So those vascular changes seem to be occurring before even amyloid starts to accumulate. So although it would be helpful to clear the amyloid and, not have it be, sitting on the vessels and causing damage to the endothelial cells, still we have to tackle the vascular alterations that happen before that. Because once we have changes in blood flow and, if there's areas of the brain that aren't getting enough oxygen and nutrients, that can cause a lot of neuronal death and can contribute to cognitive decline as well.
Yeah. Wow, that's pretty interesting. So, as a PhD candidate and graduate researcher, what are some of the biggest challenges that you're kind of faced with? And because I think I don't think a lot of people understand it's a long journey from undergraduate to graduate to a PhD candidate, and then you have to do your postdoc work.
Exactly.
Tell us about that journey and what are some of the challenges, especially as you look forward.
So definitely doing a PhD in a lab that focuses on an aging disease, I think that itself is very challenging because one of the biggest challenges is my model. We age them to 13 to 14 months, and we also have cardiovascular risk factors on top of that. So a lot of the animals don't survive. So getting enough numbers to actually, be able to have enough power to have significance in my experiments with the animals is challenging. Also, just studying the brain in general, like we said, everything is very interconnected. A lot of pathways overlap. Again, the vasculature is very complex. You have different types of vessels in different areas of the brain. It's really hard to sparse apart, what's happening in this area of the brain compared to, another area of the brain. And then also just funding is always a challenge as well, especially as a PhD student. You don't really have, too many options. You could get like a fellowship or look for outside scholarships. So that's challenging, especially because they decrease the NIA pay line for aging research. So it's very challenging, not just for me because I'm not submitting grants right now, but for, my PI, it's stressful.
Yeah. They just did that too, didn't they? Just this past year.
Yes, exactly.
So that means less money next year
exactly
for this area.
for dementia research, which is problematic.
Yeah, that is very problematic. I'm kind of curious and I don't know if you mentioned your animal model, but do you deal with tissue samples from like the brain bank to do that? And is those slices, if you would, of the brain, do they show enough that you can use that to understand that vascular system and that blood-brain barrier?
Yeah, I've never gotten samples, human samples. They're really hard to come by. We do have some just like control samples for, Alzheimer's and cerebral amyloid angiopathy, which is that's the more vascular deposition of the amyloid. So I have run some experiments with those, and they do give some interesting information. But for my project, I was trying to find also like dementia patients that had cardio risk factors as well to kind of get a little bit more translational with the results. But I don't do this, but another person within our lab, she actually collected blood samples from dementia patients, and she's seeing if they have these different cardio risk factors, and she's measuring different biomarkers within human patients. So that could reveal, like I was saying, a biomarker to screen possibly.
Yeah.
So it's going to be interesting once she finishes.
Yeah. And when you said, the brain tissue is hard to get a hold of, it's just, I don't think people realize how expensive it is to even get some of that. Is that what you're talking about?
Yeah. And the availability. People are, constantly, like you're on a waiting list basically, because I feel like there's not that many brain banks. I feel like there's less brain banks compared to other organ, especially like if you're keeping specifically Alzheimer's brains. So I know there's a few around the US, but yeah, definitely availability and then also money for sure.
Yeah, and you talked about funding, and you're a Toffler Scholar. You got a grant for that. How not to plug the too much on the trust, but how important was that to kind of helping you? Because it was in the world of grants, it was pretty small, but I think pretty important.
Yeah, for me, it definitely it helped me. I was able to order a bunch of kits for this machine called the Samoa. It's a very like high tech biomarker screening machine with high sensitivity. We just got the new one, so I was able to get a lot of kits. So I'm going to be running those. Also, I was able to travel to the Alzheimer's Association conference last year with that, which was awesome. And I got to network and share my results and get some feedback and stuff. But yeah, and I'm definitely probably going to also trying to do RNA sequencing as well. So that's something that's more on the expensive end. So saving that for that experiment as well. So that it's very helpful for sure.
Yeah. And what is the RNA sequencing that you're going to do? What are you trying to answer with that?
Ideally, it would be nice to do single cell sequencing where I could focus specifically on RNA changes within the endothelial cells. But if I could just take an area of interest in, Alzheimer's disease, for example, like the hippocampus, and sequence all the genes in that area and compare it between, my wild type and my Alzheimer's mice, and then see how the risk factors affect gene expression. It could lead to, it's really good for pathway analysis. They can group things together and kind of show that like this pathway is being really affected by this. So it's just it gives you a lot of information in a very quick, efficient way.
So what are we talked about a little bit about you're in that, in between kind of the cardiovascular system and the nervous system, and you work within that. What are some of the other kind of interdisciplinary connections or collaborations that you work with? Do you have people in your lab that you're working with like big data or computational experts, or what is that kind of collaboration that inside the lab and inside your projects?
Yeah, so there one big collaboration is there's one person in the lab who is in charge of all the behavioral testing of the animals. So this is a really important part of our research because we want to not only see the actual molecular changes, we also want to see how that translates into an animal's capability of learning a task. So that's very important. Also, we do have someone who's in charge of all the biomarker studies. So that's also important for like the vasculature, just to see what's going on within the blood circulating through the body. Yeah, mainly just like cardiovascular and neuroscience. Also, we collaborate with the lymphatics department as well, which is becoming more relevant for my project because it seems that within the TG2576 AD model mouse, it seems that there's also lymphatic deposition of amyloid. So the amyloid's also getting stuck in the vessels that not in the brain, but surround your brain and help clear waste from your brain. That's kind of what that system is in charge of. So that's also
that definitely is an emerging area of study, the lymphatic system.
Yeah, for sure.
Okay.
It's very interesting.
And does that also have the relationship to the inflammation and of, inside the brain or around the brain? Okay.
Yeah, it can. So that system is in charge of circulating cerebrospinal fluid. And then through that, actually, as we sleep, the lymphatic system is working to clear out any like debris or garbage in our brain, basically. That's why they say sleep is so important, because for multiple reasons it is. But yeah, so that's becoming another big focus. So not only you have the vasculature in the brain, you also have these other vessels that aren't, carrying blood. They're carrying fluid around your brain, basically.
Yeah, I had, I think his podcast came out today, Dr. Alan Avidid from UCLA, who's a sleep expert. And, there's a direct, he's found a direct connection between REM disorder, which is that kind of that sleep, and Parkinson's. And,
oh, wow, yeah.
Yeah. So you're right. Sleep is really important. We don't tend to get enough of it sometimes. especially as a graduate researcher, you don't probably get a lot of sleep.
No, that is very true.
So in your opinion, what are some of the kind of the most effective ways to raise public awareness and support research? You talked about, especially in Alzheimer's research and funding. You talked about NIA kind of cutting its funding. You've talked about how hard it is to get funding for you and for your PIs. What do you think, especially from maybe the younger generation? You speak for a kind of a different cohort. What do you think, we could do to kind of raise that awareness?
I think definitely like just open platforms. I think this podcast is a great idea. Open platforms where anyone can listen and, learn more about these diseases, just the general public, just to raise awareness that, especially like, for example, dementia is a huge problem now, but in 50 years, it's projected to, quadruple the amount of cases. So just to show that this is going to become a worse public health issue, and we should maybe be diverting some funds over to the aging research kind of situation. So yeah, I think just this, and also going to scientific conferences and sharing ideas and thoughts with people at the conferences. It's not just researchers; it's also other types of people that could provide you with funding, perhaps, or collaborate with you. So definitely just talking to people, I think, and just raising awareness that way. I think one of the better ways to do it.
Yeah, no, I think that's good. I've heard from several people that Alzheimer's and dementia, the more we learn, the more we know it starts way early in our lives, and that it may not be just. It is the active aging, but not for the aged. And I'm just really curious, because you're in there and you're seeing things and you're learning things about, kind of as you said, the behavior of life. Has, being in the lab, does this ever motivate you to get out there and exercise more, or, take care more, eat less salt?
Definitely does. Especially I'm like thinking about like vitamins. I'm like, oh, I hope I get enough of these vitamins because these are important.
Yeah.
No, it definitely makes me think about my lifestyle, just like my exercise, my diet, trying to not obtain, hypertension in midlife. So I don't, increase my risk, because that is for sure a well-established connection. But yeah, just it's good because like reading about it helps me with my research, but also in my own life, I like to try to at least implicate a couple of the things I learned.
Yeah, it would probably be kind of scary to like, oh
Yeah,
no, that yeah,
sometimes I wish I
exactly.
didn't know.
Yeah, right, right.
I'm like, oh,
Yeah,
too
that's
late.
so. I know you're in Sylvia's lab there, and she's one of your mentors. How important is someone like her? And I'm sure you've had other really amazing mentors. How important is that to you? Being a mentor, having good mentorship. What can you talk a little bit about that?
Yeah. Yeah. Having a good mentor, especially during your PhD, is very important. Sylvia's very busy, but if like anytime I need help or a question, she always makes herself available. But, she really has taught me like basically everything I know about the laboratory. I really didn't have that much experience, starting a PhD right out of undergrad. But no, if I didn't have a good mentor, this process would have been a lot more difficult for sure. But yeah, it just kind of shows me like what kind of scientist I want to be. And then eventually when I've mentored some, younger students, but when I'm in her position, maybe one day I want to model myself after that. So
Yeah. So what advice do you have to those people that are kind of a few years behind you? They're wanting this career.
Mhm.
They've made that decision to move from marine biology to studying the brain. What kind of advice would you give them now in hindsight?
I would have definitely started earlier getting research experience, because if anything, through the whole applying process for my PhD, like it was very like experience driven, like what have you done so far? And, I didn't do too much. So I was a little nervous about that. So definitely if you want to do research, I would say definitely start early, apply for internships, get your foot in the lab to see if you actually like it. Because it's not just it's a lot of different things, throughout the day. Sometimes you're analyzing data at your computer, and then sometimes you're running around like doing 10 experiments at the same time. So it's like to see if you actually like it, and also take the time to like go through all of the neurodegenerative diseases to see which one piques your interest. Because although they have commonalities, there's a very long list, and they all have, different outcomes. Some affect your cognition, some your motor. So just take a peek around and see which one interests you the most, I would say.
And what was the interest in the influence for you for Alzheimer's? What was that? Did you have a family member, or was there? I mean, what
Yeah.
was kind of the personal that you landed on that disease?
Neuroscience in general has that significance because several of my family members had some neurological issues, whether it was mild cognitive impairment, as they were fighting a different disease, or another one of my family members actually had cancer and it spread to the brain. So I started to, really look into neuroscience after that, because I wanted to know what was going on, because I was, old enough to, be curious and want to know. So that kind of pushed me towards that. But Alzheimer's disease in general, I don't know. I never really studied it at all. It was very novel to me. And Sylvia really did a great job explaining like kind of the background of it. And I was just very intrigued because there's a lot of answers that are missing and questions to be asked. So I thought it would be a good, field for me to do my thesis in.
So as you kind of complete your PhD, because when do you defend? Because you have to defend your thesis, correct,
Yeah.
in your paper? Yeah.
Yeah.
So when do you
September
defend?
4th of 2024 this year. And then,
So very
yes,
soon.
very soon. So I'm writing right now.
Yeah. So what are your long-term aspirations? I mean, maybe it's not a place where you want to go, but maybe there's just something, maybe a focus that you want to continue. Like, what are your aspirations, your long-term aspirations?
I definitely, so once I defend, I'm going to stay with Sylvia for a bit and finish up this in vivo portion of my project. But really long term, I'm open to working for a company, but I really want it to be neurodegenerative disease based. But also, I'm not opposed to academia either. I think I might try to apply for a grant while I'm a postdoc in her lab and see how that goes and see if I can get some funding. But long term, I would like to keep studying Alzheimer's disease and especially the vasculature. But I also wouldn't be opposed to, taking that vascular component and moving to a different neurodegenerative disease either, just to switch up my knowledge, kind of learn more about a different disease.
Yeah. Can you just kind of elaborate a little bit? Because you're at these, these pretty big fork in the road. And, some would say, if you've just spent all this time in academia, well, why don't you just stay in academia? It's, could you just elaborate a little bit about maybe the thought process or the decision making that goes, through somebody's head when they're, they're at this, because there's challenges in all the different pathways, right?
Either way, exactly. Yeah, because industry and like working for a company, there's not too many jobs open right now. It's way harder to get a job. But academia, everyone's looking for postdocs, looking for faculty positions. But then in terms of that, you have to then, once you get hired as a faculty position, you have to then keep writing grants. And if they keep decreasing the pay line, it becomes even harder and harder, to make that percentage to, get funded. So it's kind of like, it's a hard decision to make. I just really want to find somewhere where I like what I'm studying and, I like the techniques they use and kind of like the overall goal of, the project.
Yeah, it's kind of crazy to think it is something so important for us that impacts almost everyone, whether or not we get it ourselves or we have family members, that it's so hard to keep people in the positions to be able to learn and to understand and to solve these problems, because that just it just really just kind of blows me away that we don't know that much about the brain. We really don't understand, but yet and so there's just a lot of information, and we need people like you and those that are in Sylvia's lab to figure this out.
Exactly.
Yeah.
There's a lot to be found out for sure.
So when you look out in the next 5 or 10 years, what do you think some of the, besides maybe the funding and such, what do you think are some of the greatest, challenges or the greatest opportunities in this area of neuroscience and understanding Alzheimer's and dementia? Where do you, is it technology? Is it policy changes? What do you think are some of the challenges and opportunities?
I think definitely technology for sure, because, most of the time, like people don't actually get diagnosed with Alzheimer's disease until they pass away, because you have to do the postmortem analysis and confirm, because we don't have really anything that could, scan the head and say, oh, there's amyloid there. So just really like new screening techniques. Like if we could catch some changes starting to happen, like, some vascular changes, maybe some decreases in blood flow to specific areas, and we could track that with like FDG PET or something like that. I think that really like is like a main focus for sure. And a main challenge, because just being able to detect earlier instead of, waiting till a person's in their 70s or 80s and there's really nothing we could do, when the disease has progressed that much.
Yeah. Yeah. And I think about these, these blood tests too, that, you and others are kind of working on. And I think that would be pretty amazing that you draw the blood when, you're born, and every year you go in for your annual.
It's just part of the panel. Yeah. It would be
part of the panel, and then you can understand what's happening, Yeah, that's amazing. So when you think about your kind of professional goals, and what is your greatest hope for the future for you? You said, you're open to things, and you want to work on interesting things, but what are your greatest hopes, and what do you kind of hope to achieve? When you're either running a lab or running a line of business, what are those as aspirational things for you?
I definitely just want to make a positive impact, within the field of neuroscience. I want to, contribute in some way to, either thinking of or finding some pathway we could target for maybe a new therapeutic opportunity, or again, finding some specific molecule we could screen for, to catch these diseases earlier in life. So if I could just make any specific contribution to that, I think that would be really, really just my main goal.
Yeah.
And just enjoying my project also.
Yeah. Yeah. That's a big part of it, right? Yeah. It's so hard. You got to enjoy it. But I think you're already making an impact. You're publishing paper, you're doing good research, you're working with other people.
Yeah.
That's pretty amazing. So, as we kind of wrap up here, was there something that you wanted to touch on that we didn't discuss? Something that, kind of came to your mind that, additional things that you would like to maybe just kind of note in what you're doing in your research?
I think we covered most of it. Oh, I have another paper. Actually, this is one thing. I have another paper in preparation for publication right now. So that will be coming out soon. This paper is also looking at kind of the same pathways of endothelial cell dysfunction as I previously mentioned, but this time it's looking at amyloid and something called cerebral hypoperfusion. So this phenomenon is just basically decreased blood flow to the brain, to certain areas of the brain. And it's not only common in Alzheimer's disease, but also it's a consequence of cardiovascular diseases and risk factors. So for example, like hypertension is known to decrease blood flow to the brain, stroke, obviously, because it's occluding vessels and blocking blood flow. So that paper is trying to mimic that in vitro in cells. So that will be coming out soon as well, which will be exciting.
Do you know where that's going to be published? Is that going to be published in a journal, or?
Yeah, I'm thinking something more like stroke or circulation. Maybe not circulation's very high impact, but maybe some subset. Because I think the whole hypoperfusion, like disruption of blood flow, I think it would fit well into a journal like that. But yeah, I haven't picked a specific journal yet, but the paper's written, so we just have to submit it.
Well, that's great.
Yeah.
That's great. Thank you for sharing that, because I can't. That'd be exciting to read that too. Does that also get into maybe like traumatic brain injury? Does that decrease the blood flow too?
Yeah, I think that's also been shown to disrupt the hemodynamics, of the blood flow in the brain. I'm not sure about long term, but definitely acutely, like following the impact.
Yeah.
But a lot of like risk factors, even like things with the diseases with the heart, like heart failure or myocardial infarction, like all of those events have also been shown to disrupt blood flow, which makes sense because everything and like all the circulation in your body is connected. So if you disrupt one area, the brain, which requires the most oxygen and nutrients, is going to be heavily affected.
Yeah. Yeah. We don't in our normal day to day connect those things, but it definitely makes sense.
Mm-hmm.
Yeah.
Yeah.
Yeah. Yeah. More research needs to be done in that, but that's wonderful. So Ashley, as we kind of wrap up here, if people want to get a hold of you, get copies of your paper, wonder what you're going to be doing after you defend, or just learning a little bit more about what you do, how do people get in contact with you?
Definitely find me on LinkedIn. It's just Ashley Carey, Carey. Is my last name. But I think my resume is on there with my phone number as well as my personal and work email. So either way, I'll check it and find it. Yeah.
Yeah. Well, this has been great, and it's been wonderful to catch up with you. And congratulations on the 2 publications. That's pretty amazing. And good luck with your defending your your work here. And we'll check in soon to see where you land afterwards. But it's been a real pleasure, and I've really enjoyed getting caught up with you.
Same to you. Thank you so much.
Thank you for joining Research Renaissance. For more information and to stay updated, subscribe for more episodes and visit our website at tofflertrust.org. Feel free to call us if you have any thoughts, suggestions, or just want to find out more about the Karen Toffler Charitable Trust. Until we meet again, onward and upward.